Hi everyone. So today we'll be starting with the stages of tooth development. Today I'll probably start with bud stage and go on up to early bell stage, and I'll cover advanced bell stage and root formation in another video. So just giving you a brief insight. Tooth development takes place in four stages majorly. First is the bud stage, second is the cap stage, third is the early bell stage, and fourth is the advanced bell stage. What happens is that after the dental lamina starts proliferating, it forms these ovoid swellings into the ectomesenchyme, and it takes kind of this bird form, and then it takes this cap form. So these stages are named upon the morphology of the tooth germ in that particular stage. Okay, these are called the morphological stages of tooth development, and there's something called physiological stages of tooth development that I'll study later on. Okay, now moving on. This there is this line in the organs which says while the size and shape of the individual teeth are different, they pass through similar stages of development. So as we know, the incisors are smaller and the molars are bigger, but they all have similar stages of development. Okay. Now moving on, the first stage, which is the bud stage. So the bud stage starts with the differentiation of dental lamina. So this is the dental lamina, and then there's a basement membrane, and there is ectomesenchyme. So because of the interaction of the dental lamina and ectomesenchyme, the dental lamina proliferates into the ectomesenchyme, and it proliferates into 10 places throughout our arches. Okay, so we're talking about deciduous teeth formation. Okay, we have 10 deciduous teeth in each of the arch, so it will proliferate along 10 points. Each proliferation or each bud represents a deciduous tooth. So if we zoom into this bud, what we'll see is that there is the periphery is lined by low columnar cells, and the center is filled by polygonal cells. All right, and then this is the ectomesenchyme, so it shows some condensation in this stage. Okay, so the same information is written over here that it has low columnar cells and polygonal cells, and because in the ectomesenchyme there is mitosis, migration of neural crest cells, and condensation of ectomesenchyme, which will form the dental papilla and the dental sac. Okay, moving on from the bud stage, and the tooth enters the cap stage. Okay, what happens in the cap stage is that, so imagine this was your bud. There is unequal growth of the bud. Okay, it will grow from these two sides and not from over here, which forms due to the unequal growth. There is a shallow invagination. This is the shallow invagination which is formed in the cap stage. Okay, so this is the statement written that unequal growth in different parts of the tooth bud leads to the cap stage, which is characterized by a shallow invagination in the deep surface of the bud. Okay, what other features are present in the cap stage? So we had low columnar cells over here. Now this is converted into outer enamel epithelium and inner enamel epithelium. So what are outer enamel epithelium? They are cuboidal cells in here, and inner enamel epithelium, they are tall columnar, and these cells, these will form ameloblast. Okay, these will later differentiate into ameloblast, and they will, and the ameloblast will form enamel. So these are enamel forming cells. These are primitive form of enamel forming cells. Okay, what else is here? So this is the stellate reticulum. So we had some polygonal cells in here. These are converted into stellate reticulum. What is stellate reticulum? These are star-shaped cells, and how are they formed? So the polygonal cells, they absorb a lot of water. And why do they absorb a lot of water? Because we have a lot of glycosamines in the matrix of or the ground substance in here. So when they absorb a lot of water, they are converted into star-shaped cells. And this entire network, they are all connected to each other through desmosomes. Okay. So this entire network of the star-shaped cells, this is called stellate reticulum. What is the function of stellate reticulum? So they have a cushion-like consistency. Obviously, they are filled with water. So they act as a shock absorber, and they protect the enamel-forming cells. These inner enamel epithelium. This is the enamel-forming cells. So it protects them. Okay. what else is happening in outside the enamel organ? So this is the dental papilla. Okay, it was showing condensation in the previous stage. Now there is increased mitotic activity. Capillaries start developing, and some cells peripheral to enamel organs enlarge. Okay, what will happen is that you see this darkened area. Okay, the cells in here, they are going to be converted into odontoblasts. Okay, these cells, what will happen is that this inner enamel epithelium, when it's converted into ameloblast, it will send signals into this area, and these cells will be converted into odontoblasts, the dentine forming cells. So before this happens, these cells in this area, the primitive odontoblasts, they start enlarging and differentiating. Not differentiating, but they start enlarging, and there is capillary budding so that they can get nutrition. Now, and this is about the dental papilla. What is happening in the dental sac? What's happening in the dental sac? So what was the dental sac? It hasn't been formed yet, so the marginal condensation of ectomesenchyme starts. Okay, and there's more fibers and layers start to develop, and this a primitive dental sac capsular structure kind of starts developing in the cap stage. This information, it's just regarding the polygonal cells, that how they are converted into, and the stellate reticulum, so you can read that. Now moving on to the transient structures. Okay, what are the transient structures? Uh, Transient structures are seen in the cap stage and they disappear before the enamel formation begins. So in the next stage, enamel formation and dentine formation will begin. So these structures are seen only in the cap stage and they disappear. What are they? So there is the central cells of the enamel organ. Okay, cells in the central of enamel organ, they're very densely packed and they are called enamel knot. Sometimes they project outside the enamel organ into the dental papilla and they show a knob-like structure. They may act as a reservoir of cells. So they have a lot of cells and this is continuously growing. So this can provide cells to the entire enamel organ, and they also act as a signaling center and play an important role in determining the shape of the tooth. Okay, now moving on. So this will have a vertical extension. So this, I was just talking about this part. This was the enamel knot. Now this is the enamel cord. This is the vertical extension of the enamel knot. And when this extension reaches the outer enamel epithelium, it is called the enamel septum. Because at this point, it has divided the entire enamel organ into two parts. So this acts as a septum, and this is called an enamel septum. Now, upon the point of meeting outer enamel epithelium and this enamel septum, a depression is formed. This depression is called enamel navel. So these are the transient structures, and they disappear before enamel formation. Now, the bell stage. What happens in the bell stage? So, this is obviously the most significant stage, and here I'll be talking about the early bell stage. What happens is the in the bell stage, that crown shape is determined. So, until now, all the tooths, they had like a bud stage, then they all had a cap stage, and they all looked similar. But now, if I want an incisor, okay, so the incisor, for the incisor, the bell stage will kind of look like this. Okay, it will look kind of like this. If I have a molar, then it will look kind of like this. So in this stage, the shape of the tooth is determined. Okay, now how is it determined? It is determined because of the genes and their signaling molecules. So they determine which teeth is formed and where it is going to be formed. Different crown shapes is due to differential rate of mitosis and okay. So if I have a different shape, obviously this will the enamel organ will show more growth over here. So the shape is due to the different rates of mitosis and differences in differentiation. Cells begin to differentiate only when the cells cease to divide. So what will happen is, first the enamel organ will grow, okay, and then it will start forming the enamel, and then the dentine will be formed. So what it's saying, that before the enamel and the dentine, and before the ameloblast and the odontoblast are formed, first the entire growth will take place and then the differentiation of ameloblast and odontoblast will take place. Okay. The inner enamel epithelium cells which lie in the future cusp tip or incisor region stops dividing early and begin to differentiate first. So the cells in here, and you say this is the area with the incisor tip. Okay, the cells in here they will start dividing. They will start differentiating first, and this area may still start to keep growing, but these will start differentiating first. Now this diagram is a very complex diagram, but it has all the information regarding the early build stage. So you can write down your answers in these headings which I have mentioned in the form of diagram, but you can write your answers in a paragraph form and you can mention all of these as headings. So starting with what's new, we had this kind of structure up till cap stage as well. What is this? This is successional lamina, and this is new in the early build stage. So what does it represent? It represents the bud stage of the permanent successor of the teeth. Okay, so imagine if this is a central incisor, this is a germ of central incisor of a deciduous central incisor. So sometime it will be replaced by a permanent central incisor. So it is the bud of the permanent central incisor that will replace this. Okay, this is called successional lamina and it is visible in early build stage. Now there is this new layer called stratum intermedium. Okay, this layer, it is a multi-layered structure. It is formed over the inner enamel epithelium, and um, what does it do? So it has a very high degree of metabolic activity and it is essential for enamel formation. So that is the main function. It is essential for enamel formation and it is absent in the part where enamel is not formed, that is the root. So it is absent in the root portion. Now in the enamel epithelium, um, what changes are shown in this stage? So it is single layer of cell. It will differentiate into tall columnar cells before amelogenesis. Okay, so before amelogenesis, it will form tall columnar cells called ameloblast, and these are 4 to 5 micrometer in diameter and 40 micrometer in height, attached to one another through junctional complexes. So these red dots, these are attachments called junctional complexes, and they're also attached to the stratum intermedium through desmosomes. The desmosomes are also attached to each other through, the stratum intermedium cells are also attached to each other through desmosomes. Okay, they exert an organizing influence on the mesenchymal cells, which later become odontoblasts. So I'll explain this. What will happen is that these inner enamel epithelium cells, okay, they will form ameloblast, and let's just say that uh, this has formed. Now this is an ameloblast. What this ameloblast will do, it will lay an organizing influence over this dental papilla. And what will the organizing influence do? It will cause the cell of the dental papilla to differentiate into an odontoblast. Okay, so now this is an odontoblast, and this odontoblast will then lay dentine. So this will form dentine in here, and this dentine will then influence the inner enamel epithelium to form enamel in here. Okay, so this is how the whole process goes. So in that description, what's written is that basically these ameloblast will influence the dental papilla cells to form odontoblast. Now moving on, cervical loop. This is a new structure. So this is just the junction where the inner enamel epithelium meets the outer enamel epithelium. It is an area of very high intensity mitotic activity, and this will play a very important role in root formation later on. Okay, so this is where the heart with roots epithelial sheet will be formed. We'll see that later on. Now, stellate reticulum, it undergoes some major changes. What happens is that it expands further. Uh, it expands further due to more water entering the stellate reticulum. Okay, but then it collapses as well. So it collapses And what happens upon it collapsing is that this outer enamel epithelium and the inner enamel epithelium, the distance between them decreases. And it has to decrease because, as I explained to you, that dentine will be formed in there, right? So, until now, the enamel organ was getting supplied through the dental papilla, all the nutrient supply. But when once the dentine will be formed, the nutrition supply will stop. So the enamel organ will try and the inner enamel epithelium will try to derive the nutrition from outside. So to derive the nutrition from outside, this stratum intermedium will collapse and they'll come close together. So it's easier for the inner enamel epithelium to draw the nutrition from outside. Okay. And so it expands further, collapses, it reduces the distance. Okay. Uh-huh. And then what happens is that the mm, collapsing happens first near the incisor edge and then it will continue towards the cervical edge. Okay. Now going on, outer enamel epithelium. So now in this stage they are flattened to a low cuboidal form. Okay. And as I explained that it will be collapsing and this will come together. So when this will come together, it will form, they'll come closer, the outer enamel epithelium and the inner enamel epithelium, so it will be in folds. Okay, so this won't be such a smooth structure, it will become like a kind of like folding, there will be foldings in the outer enamel epithelium. And the dental sac forms papillae that contain capillary loops and provide rich nutrition supply to the avascular enamel organ. So I'll just draw a tiny diagram that what will happen to the enamel organ. So it will, so the sterate reticulum has collapsed. Okay, the outer enamel epithelium now it has come closer to the inner enamel epithelium, and the dental sac. So this portion is the dental sac. Okay, it is outside. It will kind of try to get in here. It will form papillae, and it has a lot of capillary supply. So the capillary will try to supply the inner enamel epithelium and the stratum intermedium. Okay, so this is what will happen. Now, moving to the next slide. Dental papilla. Okay, so some changes will happen in the dental papilla and the dental sac as well. What will happen in the dental papilla, as I already explained, that the inner enamel epithelium. So let's say it has organized, it is converted into ameloblast now. So uh, it will send off signals to the dental papilla, and this will start differentiating into odontoblast. So the first form of the odontoblast, it's a cuboidal form. So these cells will first take a cuboidal form, then they will form a tall columnar form. Okay, and these tall columnar, when they have the potential to secrete dentine, they are called odontoblasts. So this is how differentiation of the odontoblast will take place. There is a basement membrane separating the enamel organ and the dental papilla, and just prior to dentine formation, this is called the membrana preformativa. Now the dental sac. So before dental tissue formation, dental sac shows a circular arrangement of its fibers and resembles a capsular structure. So in the cap stage, we saw that it was showing condensation of the fibers. So dental sac, it basically forms a capsular structure around the enamel organ and a very dense structure of fibers around it. Okay. With root development, these differentiate into periodontal fibers that become embedded in the cementum and the alveolar bone. So these fibers, so right now the crown is developing. When the roots start developing, these fibers will form periodontal ligament and they will connect. They will be embedded in the cementum and the alveolar bone. Okay. So I will cover the advanced bell stage and root formation in the next video.